Imagine being told that, the absolute only way to save your life is to completely intentionally destroy your body's ability to protect itself.
Right. It sounds entirely backward.
It really does. But today, we are doing a deep dive into a stack of medical pharmacology papers that are all focused on immunosuppressants.
Yeah.
And our mission here today is to figure out how modern medicine pulls off this seemingly impossible feat. Like actually saving a person's life by deliberately turning off the very immune system we rely on to survive.
It is arguably one of the most precarious high-wire acts in all of biology, I'd say. I mean, we spend our entire childhoods learning to fortify our immune defenses, right?
Yeah, exactly. Yeah. Eating vegetables, washing our hands.
Getting enough sleep, all of that. But for hundreds of thousands of people around the world right now, survival doesn't depend on strong fortress walls.
Yeah.
It actually depends on carefully orchestrating a weak one.
Which is wild to think about. And, when I first opened these sources and saw the word immunosuppressant, my mind just immediately went to chemotherapy.
Oh, sure. That makes sense.
Right. I just pictured drugs that basically nuke the entire body to stop bad cells from growing. And the historical context in the papers points out that, medical textbooks actually used to define these drugs in exactly that brutal way.
We did. Yeah, the earliest immunosuppressive treatments were literally just repurposed cancer chemotherapy compounds.
Wow.
They were blunt instruments. The strategy wasn't to politely tell the immune system to calm down. It was to basically carpet bomb the immune cells.
Just kill them all off.
Exactly. Kill them off entirely to halt an immune response. But the evolution of this whole field is entirely about how we moved away from that widespread cellular destruction toward what we have now, which is a modern era of highly targeted pathway-specific modulation.
So we had to figure out how to silence the cells without actually killing the patient.
Precisely.
And to really grasp why scientists had to invent these incredibly complex chemical silencers, we have to look at the massive biological hurdle they were trying to overcome in the first place, which the papers refer to as the problem of self.
Right. And to understand that, let's go back to December 23rd, 1954. We're at Peter Bent Brigham Hospital in Boston.
Okay, setting the scene.
Yeah. A surgeon named Joseph Murray is standing over a patient, Richard Herrick, and Richard is in end-stage kidney failure.
And there's no modern dialysis network to fall back on back then, right?
No, not at all. He is dying. So Murray wants to try something completely unprecedented. He wants to surgically implant a healthy kidney from another human being into Richard's body.
Which normally, that is the ultimate red flag for an immune system.
Oh, absolutely.
You put a massive chunk of foreign tissue into a human, the body's defense network flags it as a huge invader, and it just tears the new organ to shreds.
It does. But Murray had a trump card here. The healthy kidney was actually coming from Richard's identical twin brother, Ronald.
Ah, right.
They were genetically identical. So
Yeah.
Richard's immune system scanned this newly implanted kidney, recognized the identical genetic markers, and just categorized it as self.
So no attack was mounted.
No attack, no immunosuppressive drugs were even needed. The surgery was a flawless success, granting Richard 8 more years of life. And it eventually earned Murray the Nobel Prize.
Which is incredible. It proved organ transplants were a surgical reality.
Yep.
But reading between the lines of the source material, this incredible victory actually triggered a massive medical crisis, didn't it?
It really did.
Because it basically proved you could save a life with a transplant, but only if the patient happened to have a spare identical twin just waiting around to donate an organ.
Right, which almost no one has. The biological wall became blindingly obvious. How do you perform this life-saving surgery between two genetically different people without the recipient's immune system just utterly destroying the gift?
Yeah.
So the race was on to find a chemical solution to bypass the immune system's really rigid definition of what is self.
And the first major breakthrough here comes from a really fascinating duo, Gertrude Edeline and George Hitchings. And they weren't even studying transplants, were they?
No, not at all. They were cancer researchers.
What?
Yeah, they were developing purine analogs. To put that simply, they were basically creating fake molecular puzzle pieces.
Okay, fake puzzle pieces.
Right, because rapidly dividing cancer cells need real purine molecules to build new DNA. So Edeline and Hitchings built counterfeit versions. When a cancer cell grabbed the fake puzzle piece, its DNA replication just broke down, and the cell died.
Oh, that's clever. And so by the early 1960s, Murray's transplant team in Boston actually takes one of these counterfeit puzzle pieces, which they modified into a drug called azathioprine, and they test it on kidney transplants between unrelated people.
And it actually works.
Yeah, it kills off the rapidly dividing immune cells just enough to stop organ rejection.
Right. And another Nobel Prize is awarded.
Naturally.
Naturally, yeah. A monumental achievement. But azathioprine was still a very blunt instrument because it interfered with DNA synthesis in any rapidly dividing cell, not just immune cells.
So it's hitting everything.
Everything. Yeah. It caused severe bone marrow suppression and left patients incredibly vulnerable. The medical field was just desperate for a holy grail, a compound that could tell the immune system to stand down without poisoning the rest of the body's machinery.
And the sources showed that the next massive leap forward didn't even come from a pristine high-tech chemistry lab. It came from a literal handful of dirt in Norway.
Yeah, it's wild. The history of pharmacology is actually heavily driven by natural products. So in 1972, scientists at the Swiss pharmaceutical company Sandoz were conducting this routine drug discovery program.
It's just business as usual.
Exactly. They were screening random fungal cultures from soil samples collected all around the world, just looking for new antibiotics.
So they test this one soil sample from I think it's the Hardangervidda Plateau in Norway.
Yep, that's the one.
And it contains a fungus called Tolypocladium inflatum. They isolate a compound from it, but as an antibiotic, it's virtually useless, right?
Completely useless. It barely kills bacteria at all. Sandoz is basically ready to throw it in the trash and move on.
But then
a pharmacologist there named Jean-Francois Borel noticed this weird anomaly in the data. This weak antibiotic was doing something profound to lymphocytes, which are our white blood cells.
Wow.
It was later named cyclosporine, and it completely shattered the existing paradigm of immunosuppression.
Because it wasn't acting like those fake puzzle pieces from before. It wasn't killing the immune cells at all.
No. Borel realized cyclosporine was potently and selectively suppressing T-cell activity.
Okay.
So the immune cells were still alive, but they were chemically pacified. It sidestepped that widespread cell-killing toxicity of drugs like azathioprine.
Let me try an analogy to visualize this shift, because this seems like where the modern era really begins.
Sure, go for it.
So the early cytotoxic drugs, those chemotherapy-style ones, were like trying to stop a break-in by blowing up the entire security building.
Yeah.
You destroy the cameras, you knock down the walls, you take out all the guards. The break-in stops, but the collateral damage is catastrophic. But cyclosporine was a game changer because it's like sneaking into the security room and just snipping the tiny wire connecting the camera to the alarm bell.
That is exactly it. The guards are still in the building, the cameras are still rolling, but the system just cannot call for backup.
Wow.
And when the FDA approved cyclosporine in 1983, it took organ transplantation from this desperate, high mortality experimental procedure and basically turned it into a standard survivable medical therapy on a global scale.
That is just amazing. Now, the source material spends a lot of time detailing exactly how you snip the wire, so to speak. They break down how different modern drugs target different specific choke points in the immune system's biological assembly line.
Yeah, and to understand those choke points, we really have to look at the immune response as a strict sequence of events, like a cascade of signals.
Okay.
So a T cell, which basically functions as a frontline general in the immune army, is patrolling the body. The moment it detects a foreign antigen, say, the tissue of a transplanted heart and alarm bell triggers inside the cell. Calcium aggressively floods into the T cell.
All right.
And this calcium influx activates a very specific enzyme called calcineurin.
Okay, let me pause you there. You just threw out calcineurin, and I know the sources mention a bunch of other proteins that follow it, like NFAT and various cytokines. Let's ground this so we don't get lost in an alphabet soup of biology here.
Fair point, yeah.
If the T cells are security guards spotting the intruder, what are all these proteins actually doing inside the guard's radio?
Okay, good analogy. Think of calcineurin as the power switch on the guard's radio.
Got it.
When calcium flows in, the radio turns on. And calcineurin's only job is to activate a messenger protein called NFAT.
Okay, NFAT.
Right. NFAT then travels down into the command center of the cell, the nucleus, and physically flips a genetic switch. Flipping that switch produces a cytokine, which is a chemical signal called interleukin-2, or IL-2.
Oh, I remember this part from the reading. IL-2 is the ultimate call for backup signal.
Yes, exactly.
It's the megaphone yelling at all the other immune cells in the area to multiply, arm themselves, and just attack the foreign organ.
Spot on. No IL-2 megaphone, no immune army. The foreign tissue is essentially ignored.
Wow.
And our dirt fungus from Norway, cyclosporine, belongs to a class of drugs called calcineurin inhibitors. It physically gets inside the T cell, binds to a structural protein, and jams the calcineurin power switch.
So the radio never even turns on.
Right. The radio stays off, the NFAT messenger never travels to the command center, the IL-2 gene is never flipped, and the megaphone stays completely silent.
That is so elegant. But what happens if a patient's body rejects cyclosporine? Like, if we know we need to jam that specific radio switch, are there other ways to do it if the Norwegian fungus just isn't an option for somebody?
Yes. So the pharmacological hunt expanded globally, and that led to a drug discovered in 1984 called tacrolimus.
Let me guess, another soil sample.
You got it. This time it was found in a sample of a Japanese soil bacterium from Mount Tsukuba.
It's always the dirt.
Always the dirt. And tacrolimus uses a different binding protein inside the cell, but it achieves the exact same calcineurin blockade as cyclosporine. In fact, it proves so potent and effective that tacrolimus is now basically the backbone of anti-rejection regimens for the vast majority of solid organ transplants worldwide today.
It just blows my mind that these incredibly specific molecular jamming devices were just sitting in dirt on opposite sides of the planet waiting to be found.
Right.
But the sources outline other drugs that work further down the assembly line, too. So what if the radio switch does turn on and the megaphone shouts, multiply? Can we still stop the attack then?
We can, yeah. We can intercept the process downstream. And this brings us to yet another remarkable geographical discovery. A drug called sirolimus, which is widely known in the medical community as rapamycin.
Oh, right. I remember the backstory on this one. It was discovered during a 1964 Canadian medical expedition to Easter Island, right?
Yes, exactly.
Which the indigenous people call Rapa Nui. They were originally taking soil samples just looking for natural antifungal compounds, and they found this incredibly strange molecule.
Yeah, the molecule from Rapa Nui operates at a completely different biological choke point. Sirolimus is what we call an mTOR inhibitor. So it doesn't stop the T cell from producing the IL-2 megaphone signal. The signal is broadcast loud and clear.
Okay, so the call for backup went out.
It went out. However, sirolimus blocks the receptor pathway, the MPOR pathway, that tells the T cells how to actually process that order.
Oh, so going back to our security analogy, the alarm bell rings, the megaphone is shouting, multiply and deploy, but sirolimus has essentially jammed the physical locks on the barracks doors.
Oh, that's good. Yes.
The troops hear the order perfectly, but they are paralyzed and cannot leave the building to execute it.
Exactly. The biological machinery required to divide and conquer is just frozen. And because sirolimus operates at a totally separate choke point than the calcineurin inhibitors, clinicians realized they could combine them.
Ah, I see.
You can attack the assembly line from two distinct angles without overlapping the exact same chemical pathways.
And the sources detail a third modern approach, too. The antimetabolites, like mycophenolate mofetil.
Yes, and the mechanism of mycophenolate is just wonderfully elegant. It deprives the immune cells of a very specific enzyme, typically abbreviated as IMPDH.
Okay, IMPDH.
Think of this enzyme as the mortar you absolutely need to stick DNA bricks together. If a cell is ordered to divide, it must build a new copy of its DNA, right? Without the mortar, replication is physically impossible.
But wait, wouldn't starving the body of DNA mortar hurt every single cell in a patient? I mean, every cell needs DNA.
And that is the brilliant part. Most other cells in the human body possess a secondary backup pathway to synthesize DNA mortar.
Oh, wow.
But lymphocytes, our aggressive immune cells, do not have this backup system. Therefore, the drug is selectively toxic primarily to the immune army, while leaving the rest of the body's tissues relatively unharmed.
Okay, I have to push back here for a second because something in the sources really doesn't add up for me.
What's that?
Well, we have spent this entire time talking about incredibly precise modern drugs. We're jamming specific radio switches with tacrolimus. We're locking down specific DNA mortar with mycophenolate. We have the biological equivalent of laser-guided missiles.
Sure.
Yet the source material notes that basic corticosteroids, like prednisone, are still a massive foundational part of immunosuppression. Weren't steroids discovered by Philip Hench way back in 1948?
They were. Hench actually won the Nobel Prize in 1950 for applying them to rheumatoid arthritis.
So why in the world, if we have these laser-guided missiles from modern laboratories, are doctors still using a blunt sledgehammer from the 1940s that suppresses hundreds of genes all at once?
It is an excellent logical question. And the answer lies in the absolute necessity of managing chemical toxicity. Even a laser-guided missile has devastating radioactive fallout if you have to build the bomb too big.
Okay. Yeah.
If a transplant surgeon relies entirely on just one precise drug, say tacrolimus, they would have to administer a massive heavy dose to completely guarantee the organ isn't rejected. But at that high dosage, tacrolimus becomes intensely toxic to the kidneys.
Oh, I see. So you hit the toxic ceiling of that specific chemical before you get the full immune suppression you actually need.
Exactly the problem.
Yeah.
So modern pharmacology relies on combination therapy. You administer a lower kidney-safe dose of tacrolimus to block the calcineurin switch. Then you add a low, safe dose of mycophenolate to starve the DNA mortar.
Right, coming from a different angle.
Right. And then you use a low dose of a broad-acting corticosteroid to generally sweep up any lingering inflammation. So by attacking the immune system from 3 distinct angles, using 3 completely different mechanisms, you achieve a synergistic, profound suppression, but you never hit the toxic ceiling of any single drug.
It's like plate spinning. You're just trying to keep all the plates spinning in the air without dropping any of them on the patient.
Exactly.
Which actually transitions us to probably the most unsettling part of this entire deep dive. Because since doctors are meticulously combining these drugs to suppress the immune system from multiple angles, what happens when they do their job, well, too well?
Yeah, you are basically forced to navigate the most dangerous trade-off in medical science.
Yeah.
A physician must suppress the immune system enough to save the transplanted organ, but not so much that the patient succumbs to an ordinary everyday infection or develops a malignancy.
And the sources bust a couple of major public misconceptions here. The first is this idea that immunosuppressants just turn off the immune system like a light switch.
Right, which if you turned off a patient's immune system completely, they would not survive the week. I mean, the environment we walk through every day is a teeming soup of microscopic threats.
Yeah.
Fungal spores in the air, bacteria on doorknobs, latent viruses. The only reason a healthy person survives a simple trip to the grocery store is because their immune system is constantly, quietly clearing these threats out. Therapeutic immunosuppression is never absolute. It is a very delicate, partial titration.
Okay, and the second big misconception is that the newer, highly targeted laboratory drugs, the biologics, are automatically safer just because they are modern and precise.
Right, and that's dangerous thinking. The term biologic simply means a large, complex molecule grown in living cells rather than chemically synthesized in the lab.
Gotcha.
They're highly targeted, yes, but targeted does not equate to risk-free.
Yeah.
The most chilling cautionary tale highlighting this reality is the pharmacological story of natalizumab.
Yes. This story really shook me when I was going through the papers. So, natalizumab is a biologic approved in 2004 for patients suffering from multiple sclerosis, or MS.
Right.
And it is incredibly targeted. Its primary function is to block immune cells from crossing the blood-brain barrier.
Yeah.
Because in multiple sclerosis, rogue immune cells inappropriately enter the brain and spinal cord, and they attack the myelin sheath, which is the protective coating around nerve fibers.
Right.
So natalizumab acts like a bouncer at a nightclub door. It stands at the blood-brain barrier and just absolutely refuses to let immune cells cross into the central nervous system.
Yeah.
And it was extraordinarily effective at halting MS progression.
But shortly after it hit the market and people started using it, cases started popping up of patients developing this incredibly rare, often fatal brain infection called PML, which stands for progressive multifocal leukoencephalopathy.
Yes.
And the real tragedy is where that infection came from, because they didn't catch a new germ on the subway. The culprit was something called the JC virus. And a huge percentage of the global adult population already carries the JC virus inside their bodies.
Which is terrifying.
It is, but it is latent. It is dormant. Because a standard healthy immune system locks the JC virus in a cage and basically throws away the key, keeping it permanently suppressed without you ever even knowing it's there.
Wait, if scientists knew this new biologic drug was designed to lower the brain's defenses and block immune cells from entering, why wasn't anyone checking patients for this dormant JC virus before giving them the prescription?
It's a very valid criticism in hindsight. But at the time, the medical community did not fully appreciate just how aggressively these latent viruses would capitalize on a highly specific, targeted blockade. They knew the bouncer was keeping the bad MS cells out,
Yeah.
but they didn't realize the bouncer was also keeping out the specific guards needed to watch the JC virus cage.
Wow. So it's like lowering a massive dam. You're purposefully lowering the water level to prevent a flood on one side of the valley of multiple sclerosis.
Yeah.
But as you drop that water level, you suddenly realize there are these dormant monsters lurking at the very bottom of the lake the entire time. And the dam was just silently holding them back all along.
That is a terrifying yet perfectly accurate conceptualization of what happened. Natalizumab was so ruthlessly efficient at keeping immune cells out of the brain that the brain was left utterly defenseless against the JC virus waking up.
Yeah.
The drug was voluntarily withdrawn in 2005. Now, it was later reintroduced with incredibly strict, mandatory viral monitoring protocols, but it remains the defining textbook example of a fundamental rule in pharmacology.
And that rule is that the biggest danger of a powerful drug is often what it stops suppressing.
Exactly. Opportunistic infections. Threats that only materialize when the chemical surveillance is lowered. And we must remember that this knife's edge of immune manipulation cuts violently in both directions, too. The sources explicitly detail the catastrophic 2006 TGN-1412 clinical trial in London.
Right, where researchers tried to do the exact opposite. They used an immune stimulant, not a suppressant.
Yeah, they administered a novel biologic antibody to 6 healthy volunteers, and it was designed to directly bind to and activate T cells. The intention was to boost immune responses safely.
Right.
But the researchers vastly overestimated the control they possessed. Within hours of the infusion, the volunteers suffered a massive, life-threatening cytokine storm. Their bodies went into multi-organ failure triggered by runaway immune overactivation.
Oh, my God.
It stands as a stark testament to just how finely tuned and how incredibly dangerous tweaking the human immune system is in either direction.
You know, if you're listening to this and thinking, well, this is fascinating, but I'm not on a kidney transplant list, so why does this actually matter to me? The sources reveal that this exact balancing act is the hidden infrastructure of modern medicine. It is happening inside millions of people you interact with every single day.
Oh, completely. The application of these pathways expands far beyond transplant wards. Tens of millions of people worldwide rely on these exact same mechanisms and often the exact same drugs to manage autoimmune conditions.
So we're talking about diseases like severe rheumatoid arthritis, Crohn's disease, lupus, psoriasis.
Yes.
Conditions where the immune system just gets confused and decides the body's own healthy tissues are foreign invaders that need to be destroyed.
Right. In an autoimmune disease, the enemy is not a surgically implanted organ. The enemy is the patient's own joints or their own intestinal lining. But the chemical solution is fundamentally identical. Dampen the immune response.
Wow.
Before the late 1990s, many of these conditions were severely debilitating. Then, a new class of biologics called TNF inhibitors became widely available.
And TNF is another one of those chemical messengers, right? Like the megaphone we talked about?
Precisely. Tumor necrosis factor is a protein that fuels the inflammatory fire, so biologics intercept that messenger before it can cause joint swelling or tissue damage. Targeted immunosuppression allows millions of individuals to live active, symptom-controlled lives today.
So just to put this in perspective for everyone, if you see a commercial for a modern biologic drug for severe psoriasis or Crohn's, or you have a family member taking one, those patients are essentially walking around society utilizing a highly tailored version of a transplant drug.
Yes, at a molecular level, the underlying mechanism is shared. They are deliberately blunting a specific aspect of their immune surveillance network to maintain their mobility and quality of life.
Which sparks some really intense ongoing debates in the medical community, like the steroid-sparing debate.
Right.
Because those old-school sledgehammer steroids, like prednisone, can cause severe bone loss, cataracts, and diabetes over the long term, so doctors naturally want to wean patients off them.
Right.
But critics argue that removing the steroid safety net raises the risk that the body will suddenly reject the organ or suffer a massive autoimmune flare-up.
It is a perpetual, lifelong recalibration, which makes the phenomenon of operational tolerance so deeply fascinating to researchers.
Oh, this part of the research just blew my mind.
It's amazing. It is incredibly rare, mostly observed in liver transplants and occasionally in kidney recipients. But a tiny, unique subset of patients can actually be meticulously weaned completely off all their immunosuppressant medications, and their immune system just accepts the foreign graft.
It just accepts it.
Yes, it defies the absolute golden medical rule that anti-rejection medication must be a lifelong commitment.
It's like the holy grail of transplant science.
It truly is. However, immunologists still cannot fully agree on why operational tolerance occurs in these specific individuals, nor do we have reliable biomarkers to identify those lucky patients in advance.
That's a shame.
So therefore, for the vast majority of patients globally, stopping their medication is just a guaranteed path to organ rejection.
And society as a whole got a really stark, real-world lesson in this biological trade-off during the COVID-19 pandemic.
We did, yeah. It laid bare the invisible vulnerability we've been dissecting today. Transplant recipients and autoimmune patients taking high-dose steroids or B-cell depleting biologics like rituximab were immediately classified as profoundly high risk.
Because to get a vaccine to actually work, you need your immune system to recognize the harmless vaccine material, build a memory of it, and manufacture antibodies.
Right.
But these patients are literally swallowing pills every morning designed to halt that exact assembly line from turning on.
It resulted in a severely blunted vaccine response, and it perfectly encapsulates the quiet burden millions carry every day. You know, the medication that keeps their transplanted heart beating or allows them to walk without agonizing joint pain inherently leaves the fortress gates cracked open to a global pandemic.
It is just an incredible scientific arc when you step back and look at the whole picture. I
Yeah.
mean, from Joseph Murray and the identical Herrick twins in a Boston hospital in 1954, proving transplants were surgically possible, but realizing we desperately needed a chemical key, to Gertrude Alain and George Hitchings repurposing toxic cancer drugs just to pry the door open, to scientists digging through soil fungi in Norway and mounting expeditions to Easter Island to discover microscopic, laser-precise molecular jammers.
It's quite the journey.
We essentially scoured the dirt of the entire globe to find the exact tools needed to tame our own cellular armies.
We spent decades learning how to decipher the internal language of the immune system, and more importantly, we learned the specific biochemical commands required to tell it to stand down.
And yet, as the story of the MS drug nitalizumab showed us with the JC virus, our mastery over this system is an illusion if we ever forget what the immune system is actively doing for us when we aren't looking.
Exactly.
Which brings us to one final thought to leave you pondering today.
Yeah, think about this. If our immune systems are expending energy 24 hours a day, 7 days a week, keeping latent, dormant pathogens permanently locked in a cage, what other hidden silent battles is your body fighting and winning right at this very second, entirely without you knowing it?
The monsters are always lurking at the bottom of the lake. We just have to hope the dam holds.